Usually when we talk about a medical diagnosis, there's this expectation of precision, like engineering.
Right, like a broken bone or something.
Yeah, exactly. You break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is, broken or not broken. It's clean.
But when you step into the world of chronic skin conditions, suddenly that x-ray machine is completely useless.
Right, because we aren't looking at a simple structural failure. We're looking at this highly complex, completely invisible ecosystem.
Which is incredibly frustrating for anyone dealing with it. Because, the symptoms are loud, the inflammation, the pain, but the root cause is entirely microscopic.
So if you've ever struggled with atopic dermatitis, or eczema, as most people know it, you know exactly what that frustration feels like. You've got the maddening itch, the severe dryness, that feeling that your skin barrier is just, fundamentally broken.
And you've probably blamed your genetics, right?
Right.
Or the dry winter air, or maybe even a new laundry detergent.
Oh, I've definitely thrown away detergent thinking it was the culprit. But we're doing a deep dive today into a groundbreaking 2026 paper from Cell Reports Medicine that it shifts the blame entirely. Our mission today is to unpack this because it points to a massive communication breakdown between your human skin cells and the bacteria that live on top of them.
Yeah, the mechanism they uncover here changes how we view the skin entirely. We've known for a long time that in atopic dermatitis, the skin microbiome is out of balance.
A state of dysbiosis.
Exactly. The pathogenic bacteria, specifically Staphylococcus aureus, multiply rapidly. Meanwhile, the beneficial bacteria, specifically the genus Cutibacterium, just decline.
So the bad guys are storming the gates and the good guys are retreating. But what this paper asks is the crucial next question, which is, why do the good bacteria fail? Like, what is actually happening on a cellular level when that defense line collapses?
To understand the collapse, we really have to look closely at the primary defender, which is Cutibacterium acnes, or C. acnes.
Now, I have to stop you there, because when anyone hears the word acnes, they immediately think of teenage breakouts. Like, this bacterium has terrible PR.
It really does.
I mean, we spend millions of dollars a year buying products specifically designed to eradicate it.
And it's arguably the most misunderstood microbe on the human body. That reputation is largely unfair because C. acnes is actually the most widely distributed bacterium on normal, healthy human skin.
Wow, really? So it's supposed to be there?
Oh, absolutely. Think of it as a cutaneous caretaker. It's doing a tremendous amount of heavy lifting for your immune system.
Right, so it's not just hanging out, it's actively terraforming the environment.
Exactly. It lowers the pH of our skin, maintaining this acidic environment where bad pathogens like S. aureus struggle to survive. It also promotes the secretion of antimicrobial peptides.
Okay, so it's actively fighting.
It is. But its most powerful defense mechanism involves your natural skin oil, or sebum. C. acnes metabolizes that sebum and turns it into short-chain fatty acids.
And those fatty acids do what, exactly?
They actively inhibit the growth and the biofilm formation of the bad pathogens.
Okay, so it's taking our natural skin oil and weaponizing it against invaders, which, brings up an obvious contradiction here. If someone has atopic dermatitis, they still have C. acnes on their skin.
Right. It might be reduced, but it's there.
So why isn't it fighting off the bad bacteria?
That anomaly is exactly what drove the researchers to use whole genome sequencing on the bacteria. They wanted to see if there was a genetic difference.
Whole genome sequencing, so they read the entire genetic code of the bacteria on different people?
Spot on. They isolated C. acnes, strains from the faces of healthy individuals, and compared them to strains taken from patients with atopic dermatitis.
And what did they find?
What they found was a stark genomic shift. The C. acnes living on the inflamed skin were genetically and functionally different from the ones on healthy skin.
So they basically mutated.
Yes, there was massive strain-level heterogeneity. The specific strains dominating the skin of atopic dermatitis patients are fundamentally missing a critical ability.
Like they forgot how to do their job.
Kind of, yeah. When the researchers ran a metabolomic profile, essentially looking at all the chemical byproducts the bacteria produce, they found a glaring absence.
What was missing?
The mutated strains had essentially forgotten how to manufacture a specific metabolite called indolelactic acid, or ILA.
Okay, let me use an analogy here. It's like the guards are still standing on the wall, but they've dropped their weapons, and ILA is that missing weapon.
Precisely. In a healthy ecosystem, C. acnes creates ILA by digesting an amino acid called tryptophan.
So they eat tryptophan and they pump out ILA, which protects the skin.
Right. But in the diseased state, that production just plummets.
My immediate thought is, well, let's just fix the supply chain. If they need tryptophan to make ILA, can we just give them more tryptophan? Can we force these lazy bacterial strains to start manufacturing the weapon again?
We can try, but the researchers discovered a major biological hurdle. The bacteria's ability to produce ILA isn't just about having the raw materials. It's completely dependent on the surrounding environment.
What kind of environment?
They found what you could call a Goldilocks sebum effect.
Meaning the skin oil has to be just right.
Exactly. They tested the bacteria in different concentrations of artificial sebum. They tried 0.01%, 0.05%, and 0.25%.
And I'm guessing more oil wasn't necessarily better.
The fascinating thing is that ILA production didn't just scale up as the oil increased. It peaked only at that middle 0.05% level.
Wait, really? So if you have too much oil, like the 0.25%, they just stop making the protective metabolite?
They shut down production completely. And if there's too little oil, the 0.01%, they also shut down. The environment has to be perfectly balanced for the bacteria to feel comfortable enough, or perhaps biologically triggered, to produce this specific defensive chemical.
Okay, if you're listening to this and you're someone who aggressively scrubs your face every night, hoping to wash away every last drop of natural oil to feel quote-unquote clean, you might want to rethink that strategy. You might be deliberately creating an environment where your own security force refuses to arm itself. You're basically starving the system.
It really highlights how delicate the balance is. We are constantly disrupting an ecosystem that relies on microscopic precision.
Okay, let's do a quick 5-minute recap here to lock this in. I'm going to run through the key takeaways so far.
Go for it.
1, atopic dermatitis involves a damaged skin barrier and a dysbiotic microbiome, so bad bacteria up, good bacteria down. 2, C. acnes is normally a protector, not just a teenager's worst nightmare. And 3, in AD patients, C. acnes undergoes this genomic shift and stops producing the crucial tryptophan metabolite, ILA, especially when sebum levels aren't perfect.
That's a perfect summary.
So my question to you, the listener, is this: does this change how you view washing away all your natural skin oils? Think about that. Okay, moving into the next phase. We know ILA is missing, but I want to get into the actual mechanics of this. What does ILA actually do when it is present? How does a bacterial byproduct physically repair cracked, dry, inflamed human skin?
To understand the physical repair, we need to look at the outermost layer of your skin, which is made up of cells called keratinocytes.
Keratinocytes, got it.
Inside these human skin cells, there is a specific receptor known as the aryl hydrocarbon receptor, or AR.
AAR. Okay, can you give me a memory trigger for that?
Yeah, think of AR as the armor healing receptor.
Oh, I like that. Armor healing receptor. So what is this receptor usually doing? Is it just waiting around for bacteria to talk to it?
Well, historically, we've known AAR as an environmental sensor. It's famous for detecting external toxins, things like dioxins or pollutants, and triggering a cellular response.
That's an alarm system.
Right, it's an alarm. But what biologists are increasingly realizing is that our cells didn't evolve these incredibly complex receptors just to detect man-made pollutants. They evolved to detect endogenous natural signals.
And this paper proves that the bacterial metabolite, ILA, is the natural, perfectly shaped key for the AHR lock.
Yes.
When you say perfectly shaped, how do we actually know that? Are we just guessing because putting ILA on the skin makes it look better?
Not at all. The researchers used 3D molecular docking models to map the exact physical interaction. It's breathtaking to see. The ILA molecule enters the human skin cell and physically binds to the AHR receptor.
Like a literal puzzle piece.
Exactly like a puzzle piece. It forms 2 highly stable hydrogen bonds with a specific amino acid called serine 346, 1 bond with histidine 337, and it even engages in pi-pi stacked interactions with phenylalanine 295 on the receptor.
Whoa, okay, that is wildly specific. 2 bonds with SAR 346, 1 with his 337, and pi-pi stacking with Phe 295.
The geometry is so precise that the equilibrium dissociation constant, the KD, is 4.23 micromolar, which basically means the binding affinity, the strength of the chemical grip, is incredibly strong.
So this isn't some random chemical reaction where 2 things just, bump into each other in the dark. It is a highly specific intentional fit. Our human cells have a lock that is structurally designed to receive this exact bacterial key.
It is the absolute definition of symbiosis. But the most important part is what happens after the key turns the lock.
Right, what does it do?
Once ILA binds to the AHR receptor, the receptor physically moves. It translocates from the outer body of the cell straight into the nucleus, the control room.
And once it's in the control room?
It acts as a master switch for the skin's structural integrity. Specifically, it upregulates the production of tight junction proteins.
Give me a visual for tight junctions. What are we building here?
Imagine your skin cells are bricks. If you just stack bricks on top of each other, water will easily seep through the cracks.
Right.
Tight junctions, which in this study are specific proteins with names like CLDN1, CLDN4, and OCLN, are the microscopic zip ties, or the heavy-duty mortar, that bind those cellular bricks incredibly tightly together.
So they create a waterproof seal.
Exactly. So when that seal is broken, that's when you get the severe dryness, the water just evaporating out of the skin, which is a hallmark of atopic dermatitis.
The medical term is transepidermal water loss, right?
Yes, TEWL. The skin is literally leaking its own moisture. By turning on the master switch, ILA forces the human cells to manufacture more zip ties and seal the leaks.
Okay, that handles the dryness and the broken barrier, but what about the maddening itch and the redness? Atopic dermatitis isn't just dry, it is fiercely inflamed.
That brings us to the second function of this master switch. While AHR is in the nucleus building the structural wall, it is simultaneously suppressing the production of a molecule called interleukin-33, or IL-33.
IL-33, let me guess. This is a pro-inflammatory cytokine. The fire alarm.
Spot on analogy.
Yeah.
IL-33 is the chemical fire alarm that your skin cells release when they are damaged or stressed.
And it triggers the itch.
It floods the area, triggering massive immune-driven inflammation and that intense, inescapable itch. So the ILA from the bacteria is doing two things at once. It's rebuilding the wall with tight junctions, and it's turning off the fire alarm by down-regulating IL-33.
Just think about the implications of that for a second. The structural integrity of your skin, how well it holds water, and the inflammation levels of your immune system are being directly controlled by a byproduct of the bacteria living on your face.
It's wild, isn't it?
It really blurs the line of where the human ends and the microbe begins.
It absolutely forces us to view the human body not as an isolated organism, but as a host for a complex, interdependent ecosystem.
Okay, time for our 10-minute recap. I'm going to fire off the bullet points for segment 2.
Let's hear it.
1. ILA binds precisely to the AHR receptor, the armor-healing receptor inside our skin cells. 2. This exact 3D docking activation causes AHR to move to the nucleus. 3. This process increases tight junction proteins like CLDN1 and CLDN4, essentially microscopic zip ties to seal the skin. And 4. It simultaneously lowers IL-33, shutting off the fire alarm to reduce inflammation and itching.
Perfect recap.
So, listener, if a bacterial byproduct controls your skin's structural integrity, who is really in charge of your body? It's a trippy thought.
It really is.
Okay. The biological elegance of this is undeniable. The key fits the lock, the switch flips, the zip ties are built, the alarm is silenced. But I have to play the skeptic here.
Please do.
A molecular mechanism that looks perfect in a 3D computer model or in a petri dish notoriously fails when you apply it to a living, breathing, messy organism. How did they prove this actually heals skin?
They moved to in vivo testing. They used living mouse models, and they didn't just do one test. They used two completely different methods of skin destruction to ensure the results were robust.
How do you artificially break a mouse's skin barrier in a lab?
First, they took normal C57BL6 mice and subjected them to acute barrier disruption through a method called tape stripping.
Tape stripping, that sounds exactly like what it is.
It is. They essentially apply and remove adhesive tape repeatedly to strip away the outermost protective layers of the skin. It simulates severe physical damage and immediate water loss.
Sounds brutal, but effective for the test. And the second model?
The second was a chemically induced dermatitis on BALBC mice. They applied a specific chemical called MC903 to the ears of these mice.
MC903, and what does that do?
This chemical reliably forces the immune system to overreact, causing severe atopic dermatitis-like inflammation, scaling, and swelling.
So they have one model of pure physical damage from the tape and one model of severe immune inflammation from the chemical.
Exactly. And when they applied the bacterial metabolite ILA to these two very different types of skin damage,
what happened?
the topical application worked brilliantly in both scenarios. In the physically damaged mice, the ILA significantly accelerated barrier repair. The skin rebuilt its layers and stopped leaking water much faster than the control group.
And in the inflamed mice?
In the chemically inflamed mice, the ILA markedly reduced the scaling, decreased the visible swelling of the ear tissue, and critically lowered the levels of inflammatory markers in the blood.
But hold on. How do we know for an absolute fact that the healing was caused by that specific IIR receptor pathway we just talked about?
The scientific control.
Right, because if I put a nice thick layer of petroleum jelly on damaged skin, it also stops water loss and helps it heal. Maybe ILA is just a nice moisturizer.
This is where the experimental design shines. To prove the exact signaling pathway, they introduced a genetic knockout twist.
A knockout twist.
Yeah, they engineered a highly specific strain of mice called K14 Cree Arfel mice.
K14 Cree Arfel, catchy name.
Very catchy. But the important thing is, these mice were perfectly normal in every single way, except for one detail. Their skin cells completely lacked the aryl receptor.
Oh, they literally removed the lock.
Exactly. The mouse equivalent of the keratinocytes had no IR keyholes. When the researchers induced the severe dermatitis in these genetically modified mice and applied the ILA, absolutely nothing happened.
Wow, the ILA was useless.
Completely ineffective. The inflammation raged on, the skin barrier remained broken, the tight junction zip ties were not built.
That proves ISR is the mandatory middleman. Without IR, ILA is just useless.
Exactly. It proves definitively that ILA isn't just a physical barrier or a soothing ointment. It is a biological signaling molecule that requires that IR receptor to trigger the healing cascade.
That is fantastic science. The knockout control proves the causation. But of course, mice are not men. We've cured cancer in mice 1,000 times. If this is going to change how we treat eczema, I need to see human data. Did they take this to clinical trials?
They did. They set up a proof-of-concept clinical trial with 10 human patients suffering from mild to moderate atopic dermatitis.
10 patients. Okay, small, but it's a start.
Right, and they utilized a split-body design, which is highly effective in dermatology for eliminating variables.
Right, because if you test one person against a different person, genetics and environment get in the way. But with a split-body design, you test the left arm against the right arm of the same person.
Exactly. They identified symmetrical inflamed lesions on each patient. For 2 weeks, they applied the active ILA treatment to the lesion on one side and a vehicle control, just the plain base cream without the ILA, to the lesion on the other side.
The ultimate test. What happened after 2 weeks?
The clinical outcomes were statistically significant. The side treated with ILA showed a major reduction in transepidermal water loss, our old friend TEWL.
So the human skin physically rebuilt its barrier and stopped leaking moisture.
Yes. Furthermore, they measured the results using the SCORAD metric, the Scoring Atopic Dermatitis tool, which factors in redness, swelling, and patient-reported itchiness.
And the SCORAD went down.
The SCORAD was significantly lowered on the ILA-treated side compared to the placebo side.
It translated perfectly, from the computer model to the knockout mice, directly to human relief.
It did, and the researchers ran one final, incredibly ambitious experiment to bring the entire narrative full circle. Up to this point, they had been applying purified synthetic ILA directly to the skin.
Right, isolated chemicals.
But they wanted to know, could the living ecosystem do the job itself?
Wait, could they just apply the live bacteria to the skin?
Yes. They took a live, high ILA-producing strain of C. acnes, specifically 1 × 10 to the 7th CFU, so a very high concentration.
But remember the Goldilocks rule. The bacteria won't perform if they are stressed or starved.
You're paying attention. They didn't just apply the bacteria, they applied the live bacteria, combined with its favorite raw material, tryptophan, and that perfectly balanced 0.05% artificial sebum.
Oh, wow. They gave the microscopic security force, their weapons, and their rations, and deployed them straight onto the inflamed skin.
And it worked. The living ecosystem, when given the proper fuel and environment, successfully attenuated the inflammation and repaired the skin barrier in the subjects. It did the exact same job as the isolated chemical.
Okay, we need our 15-minute recap right here because that is huge.
Hit me.
Bullet point 1, ILA successfully reduced water loss and repaired skin in both physical and chemical mouse models. Bullet point 2, mice genetically lacking the AHR receptor saw 0 benefit, completely proving the biological pathway. 3, a 10-person human trial showed real-world reduction in AD symptoms over 2 weeks. And 4, applying live C. acnes with its favorite food, tryptophan, and 0.05% sebum also reversed the dermatitis.
Spot on.
So, to our listener, would you be willing to apply a lotion made of live bacteria and skin oil if it cured your dry skin?
That is a total paradigm shift, isn't it? Think about what that means for the future of skincare.
Right now, if you have a skin condition, you go to the store and buy a tub of inert cream, or maybe a harsh steroid that just shucks down your immune system completely.
Exactly. But what this paper is suggesting is that you could apply a living lotion, a serum made of live bacteria, specific amino acids, and synthetic oil that organically interfaces with your cells to heal the skin from the inside out.
It fundamentally changes the philosophy of dermatology. We move away from the idea of just moisturizing dead skin cells or sterilizing the surface, and we move toward the concept of farming a microbiome.
I love that phrase, farming a microbiome. In fact, in the discussion section of the paper, the authors introduce a concept that I believe will dominate the next decade of skincare science, which is cutotypes.
Cutotypes, I haven't heard that term. Is it similar to how we categorize blood types?
It's a very similar framework. Just as you have type A or type O blood, human skin can be categorized by its dominant bacterial ecosystem. For example, some individuals naturally have a C cutotype, meaning their skin is heavily dominated and protected by C acnes.
Okay.
Other individuals might have an M cutotype, where their ecosystem is dominated by a completely different microbe, like Moraxella.
Oh, which totally explains why skincare is so maddeningly inconsistent. Like a heavy oil-based moisturizer might perfectly feed the C cutotype person, allowing their bacteria to thrive and protect them. But if an M cutotype person uses that exact same product, it might cause a massive inflammatory breakout because they're feeding the wrong ecosystem.
Precisely. We've been using blunt instruments on highly specialized ecosystems. If we extrapolate this research into the near future, the way we treat skin conditions is going to become hyper-personalized.
So you won't just buy a generic lotion?
No, you'll go to a clinic, they will take a swab, sequence the DNA of your skin's microbiome, and determine your precise cutotype.
And once they know your cutotype, they build the product around it.
Exactly. You would be prescribed a custom live probiotic serum. It would contain the exact strains of healthy bacteria your specific skin is missing. It would contain the specific raw materials, like tryptophan, that your bacteria need to manufacture protective metabolites. And it would be suspended in the exact percentage of synthetic sebum required to optimize their environment.
You literally become a conscious caretaker of your own microscopic ecosystem.
We have to realize that our survival and health are cooperative. We need these microbes just as much as they need us.
It completely changes how you view yourself. We started this deep dive looking at a frustrating, painful condition, atopic dermatitis, where the skin barrier just falls apart.
Right.
And by following the science, we discovered that the culprit isn't just a lack of moisture, but a genetic shift in our bacterial defenders. We learned how they lose their ILA weapons, how ILA physically fits into our cellular locks to build microscopic zip ties, and how living, breathing bacteria can actually be deployed as medicine.
It's incredible.
So here is a final thought to carry with you. The next time you walk into your bathroom and look in the mirror to wash your face, try to see past your own reflection. Try to imagine the billions of invisible guards standing watch on your skin. You are a complex, beautiful, and delicate ecosystem, and you are responsible for feeding, balancing, and protecting it. Maybe go a little easy on the harsh chemical scrubs. Give your C acnes a fighting chance and let your cutaneous caretakers do their job.
It's really about working with your biology rather than constantly fighting against it.
Thank you so much for joining us on this deep dive. We hope you walk away looking at your own skin in a completely new light. Keep asking questions, keep exploring, and we'll see you next time.